Okay, so today in the Bitesize Bioinformatics session, we are going to take a look at SeqMonk, which is a piece of software we've been developing at Babraham for about 11 years now. It's quite old. This is a piece of software which exists to visualize, explore, and analyze large sets of mapped sequence data. So mostly this is data that comes from high throughput sequencing experiments. Anything where the point of doing the sequencing is to identify positions on a genome. So ChIP-Seq, ATAC-Seq, RNA-Seq, bisulfite sequencing, Hi-C, any of those kind of techniques are amenable to work within here. But any other data type that generates a large set of genomic positions that you want to be able to visualize and analyze would also work with this as well. The program itself is a graphical program, so it's a bit different to a lot of the other NGS tools, many of which are sort of command line driven or exist in an environment such as R. One of the things that we're going to hopefully sort of illustrate as we go through today is kind of why we go that route with the program, and it's really all about exploring and understanding your data and being able to spot things that are either really good and you can be really happy with, or a bit weird and are quite difficult to spot in a command line environment, but are really easy if you have a nice sort of visual interactive environment, which is what we have on here. SeqMonk is free software, so it's released under the GPL license, so it's available for academics or commercial people to use with basically no restrictions on its use on any kind of data without having to pay us anything or get permission to do that. And it is a cross-platform application, so I'm going to be running it on a Windows machine today, but there is a Mac version and a Linux version, and they look and work exactly the same, so there's basically no difference between them. Let's start with some basics then about just how do you get the program, set it up, and get it working. I'm going to switch across to sharing my screen then. So here I'm showing just a view of my group's web pages. So inside here, under projects, we have all of our different software projects that we distribute, and SeqMonk is under there. Okay, so you can see the page for that and download link. Actually, it's probably worth pointing out, I need to update this. So on the download page, it says requirements a suitable Java runtime environment. That's not true anymore. So we now embed a copy of Java in with this. So it's a program written in Java, but you don't actually need any dependencies to get the program working. And from the download link, there is just a binary download for either Windows, Linux, or Mac, and you just click on the appropriate one to download for your platform. On Windows or Linux, they're just compressed files, so it's a zip file on Windows, and it's a tar file on Linux that you just unzip or untar. On macOS, it's a DMG disk image, and you just open that and then drag the icon out from the image file to wherever you like on your machine, and that will install it. If I show you what you get in the Windows version, I'm just going to switch across to a copy that I've just downloaded and unzipped. This is what you get if you unzip it. So there's a whole bunch of files, but somewhere down the bottom here is SeqMonk.exe, and just by double-clicking that will start the program. Okay, so I'm going to do this. You would normally see the startup sequence on here, but again, because of Zoom, Zoom won't show you that. So I'm going to switch across to what has just opened up on my screen. That's interesting. Okay, I'm going to have to show you the whole screen briefly because some of the way that Zoom works, some of the windows that SeqMonk opens show up and some don't. So for the most part, we'll be okay, but I'm just going to quickly show you the whole screen. So on here, we have the screen that you will see the very first time that you start SeqMonk. it needs a couple of things to set up when you first start it. So one of those is the location of a couple of folders that it uses when it runs. So the two folders that are on here, one is just a cache folder. So that means that when it's running, it needs to temporarily write out a bunch of data to somewhere on your machine and load it back in. Nothing is permanently stored there, but it needs to temporarily store stuff there. So you need to set a folder for that. The default is inside your home directory, but if you want to put it somewhere else, you can. The other folder that's in there is the genomes folder. We'll see in a second that SeqMonk has to download a whole load of genome annotation information, and again, it just needs to know where it can save that. So if you're happy with both of those choices, you can just start using SeqMonk. There's a couple of other bits of setup to do. So the other thing that you need to do right at the start is you for some of the functionality, not all of it, but for some functionality in SeqMonk, we link out to a local installation of R. So as well as having SeqMonk, it's useful to install R on your machine. You can just get that from cran.rproject.org. And then inside SeqMonk, you'll get something where it says it couldn't find a default R installation, but there's a button to say detect R. We press that, hopefully it should find it. If you can't, then it will prompt you to actually go and locate it. But in here, it found my R install already. It will then check the R installation, and all it's doing is to look, firstly, that it works, and secondly, that it has a specific set of packages that SeqMonk uses itself. So here, it did the check, and it found that it hasn't got all the packages that it wants. So there's a button again to install the R dependencies. So if I do that, it should now go off and install the packages that I'm missing, and then it should be set up and running. It has to be said, this is by far the flakiest part of installing or using SeqMonk. Our installations are not robust. So it should work every time, and R should find all the dependencies. But if it doesn't, you'll get an error message. And if you submit that error message to us, we will reply to you and tell you why it didn't work. But nearly all of the problems that we see reported to us in SeqMonk end up just being R dependency problems. One thing that has definitely turned up in the last few months is that for some of the packages that we use, the R repositories have been completely broken for any version of R less than R 3.6. So if you're using a 3.5 or a 3.4 release of R, it just won't work anymore. The package repositories are just fundamentally broken. So for SeqMonk, for the packages that we use, you have to use R 3.6, which is the latest release, which is what you should be using anyway, but that should get you up and running. Okay, so once we've set those cache directories and we've created the link to the local install of R and installed the packages, we should get a nice little set of green ticks down here, and we're okay to start using the program then. So let me just expand that view. Okay. So let's go about starting to use the program, importing some data. I've got a couple of example data files that I'm going to bring in and play with. Again, I'm not going to say much at all about what these data files are, because for today, it doesn't matter. I'm sort of illustrating the function of the software and not trying to get too much into the details of the biology of the type of data that we're working with. The input that you're going to bring into SeqMonk will be a set of mapped genomic positions. Normally, this will be in the form of BAM files. So these will be things that have come out of a high throughput aligner, so things like BWA or Bowtie 2 or HiSat 2 or STAR or any of those programs that do that. They all use the same file format. The starting point for working with those in SeqMonk is that you have to create a project. So creating a project is just file and new project. And the basis for a project is a particular version of an assembled genome, or assembled and annotated genome. So what you see in here is the list of genomes that you have installed on your machine. Now, if you've just installed SeqMonk for the first time, this list will be completely empty. You won't see anything in here at all. I've got I've got a few genomes that I've installed before, so I can see them, but I'm going to go through and show you what the installation of a new genome would look like. It is critically important that the genome that you select for starting your new project is the same genome that you mapped your data against. Because in the end, what your BAM files provide are a set of genomic positions. They say this read goes on chromosome 3 from position 500 to position 600. Okay? And those numbers only mean anything in the context of a very specific genome assembly. Those positions will exist in other assemblies, but they won't line up against the same genes and they won't mean the same thing. So you have to be sure that the genome that you're using here is the same as the genome that you mapped against. If you're not sure which genome you mapped against, you can look in the header of the BAM file. So programs like SAMtools can be used to view the header of the BAM file, and that should tell you what command was used to map it, and the command normally contains the name of the genome. But if you're not sure, find out from the person who mapped it what that genome was. The data that I'm going to import was mapped against the mouse genome, and it was mapped against the GRC-M38 version of the genome. So I'm going to go off to our server and bring down a copy of the GRC-M38 genome. So here, let's pretend this was completely empty, and I can just say import genome from server. This gives me a list of all the genomes that are on our server, and if I go to M and scroll down to mus musculus and GRC-M38, you'll see a number of different versions in here. Genome assemblies come in kind of two parts. So here you can see things where it says GRC-M38 and then V90, V95, and V97. The bit that matters for matching to your BAM file is just the first part of this. This is the GRC-M38. Okay, so GRC-M38 is the name of the actual assembly, the order of bases that you get in your chromosomes, okay, and the thing that determines the coordinate system. The number on the end, the V90, V95, and V97, are releases of the annotations that then go on top of that assembly. Okay, so all of our genomes are drawn from the Ensemble project, and Ensemble re-annotate their genomes fairly regularly. So for the same underlying sequence and therefore the same mapped positions, you can have newer and newer versions of annotations where the gene models have been refined and names updated and all of that sort of thing. So if I've mapped my data against GRC-M38, I could select any of these and they will all work, but it's normally a good idea to pick whatever the newest version of the annotation is, because that's going to be the most recent information. Okay, so I shall download that. And once it's downloaded, it will then take me back to the open project. And now if I look under mus musculus and GRC-M38, I had the 95 version installed before, but now I've got 97. Okay, so I've selected my correct genome version and I can start my project. The first time you start a project with a new genome, it'll take a little bit of time just to start up, but for every other launch after that, it'll be instantaneous. And then the main interface for the program should open up like that. Okay, now before I get into actually looking at the interface itself, I'm going to import some data. So I've got a couple of BAM files that I'm going to import. As it happens, these are ChIP-Seq BAM files. They're histone ChIP files, but again, I'm not going to get into the details of that, but just to sort of give you the idea of what it is you're going to see. Bringing in data then, once you've created a project, is done by File and Import Data. SeqMonk supports import from a lot of different standard formats of file, but most of the time you're going to be using BAM files. So here I'm going to select BAM or SAM. I can then find the files that I'm going to import. So I'm just going to go into the folder that's got them, and I'm going to import both of these files. Okay, so you can import as many BAM files in one go as you like. They just have to be imported with the same settings, which you should be doing anyway. So here I'm going to import both of them. There are options that you get to set when you import your BAM files. Normally, the program has looked inside the BAM file and set the options correctly, but it's always worth just having a quick check down. So I'm going to very quickly go through what the options are on here. Remove duplicate reads. So if you have more than one read in exactly the same position on the genome, sometimes that can be an indication of PCR duplicates. It's not something you should generally do just as a matter of course, and I'm certainly not going to do it here, only if you know you have a problem with duplication should you think about doing that, but I'm not going to do that. Hi-C is a particular data type that is used for measuring genome structure, and it's a data type that you can't automatically detect by looking at the BAM file. Hi-C BAM files look like lots of other things as well, so we can't figure that out for ourselves. So if you have a Hi-C data set, you need to tick the box, but if you don't, then you don't. And then there are a couple of Hi-C options, specific options, which we're not going to get into. One option which is useful to know about is the mapping quality. So inside a BAM file, when you report a hit, you say, well, this read goes to chromosome 3, position 400 to 500. It will give a numerical value for how confident the aligner was that that position is correct. And that is driven mostly off how unique the match is. So if there was only one place in the genome that was a good match to the sequence that you were looking at, then you will get a very high mapping quality score. And if the read that you've got could have mapped to lots of places in the genome, you'll get a very low quality mapping score. So we, by default, filter for a mapping quality of 20. A mapping quality of 20 means that the reported position should be 99% certain of being correct. And that's the way these numbers work out. But basically, all it does is to kick out all of the reads which have multiple places that they could have mapped. So we're only looking at uniquely mapped reads. Related to that is the one underneath, which is primary alignments only. Again, depending on the settings that you've used for your alignment program, if you can tell some alignment programs to allow it to report multiple genomic locations for the same read. If it does that, one of them has to be flagged up as the primary alignment for that read. So if there's one that's better than the other, that will be flagged up. If there's several that are equally good, then just randomly one of those will be picked. But again, by default, we only allow primary alignments, which means we never should import more than one mapped position for one of the original sequences. If we're filtering on mapping quality, this really wouldn't do much because normally you wouldn't have any multiple reported reads anyway, but you can make your aligner do stuff like that. So it's safer to do it that way. Treat as RNA-Seq data is a difference in the import which we do if you have spliced alignments in your read. So basically we split the reads apart. So you look at the exonic portions. SeqMonk will figure out from your data if it is spliced and will tick that for you already. But in this case, it isn't, so it hasn't. Finally, it will look at the type of library. So there are two types of library that you can do, or two types of sequencing you can do on most sequencers, which are either single-end sequencing or paired-end sequencing. And this simply says that if you have an insert that you're sequencing, you can either sequence it just from one end. So you, oh, my fingers don't appear on the screen. There we go. So you can either sequence it from one end, or you can have a pair of sequences where they sequence from opposing ends, and you get two. The program will figure it out. So generally, you can just leave that exactly as it is. And then at the bottom, there are options that come for different types of library. So in this one, it's a single end library, so you get the option to extend the read. And I'm not going to go into that today because we'll talk about that if we talk more specifically about ChIP data. So I'll just go ahead and then start the import. And it's going to read my two BAM files and then import them. This might take a little bit of time. BAM files are big, but you can see it's loading them pretty quickly. Each little tick that's going up on here is 100,000 reads coming in. So it is reading this data stupid quickly. But data sets these days can either easily have 20, 30, 40 million reads per sample. So you might be easily importing several 100 million reads into your data set. So this can take a few minutes to do. Once you've imported the data from the BAM files, then SeqMonk actually keeps a copy of everything that it needs. So you don't need the BAM files to stay around on your machine once you've done the initial import. Okay, so once we've done this, SeqMonk has got its own copy of this, and you don't need to keep referring back to the original files. Okay, so now we've started a project and imported some data, we can have a look around the interface that we're working with in SeqMonk. We've got three main windows. The top left is what's called the data view, and it's really just the status of the project, showing you what you've got loaded and what you can work with. On the top right, you have the genome view, which is kind of a sort of carrier type view of the genome. And then in the bottom, we have the chromosome view, which is a track-based genome and data browser, which is where most of the interesting stuff happens. I'm going to quickly walk you through the different windows and show you what's in there, and then more things will sort of populate themselves. More things will populate themselves as we start doing more things inside the program. Okay, so let's start with the data view. Inside here, we've got a series of folders. At the top, it tells us what genome we're using. So this is just telling us this is the mouse GRCM 3890 version 97 genome. Underneath that, we can see annotation sets. So we get a bunch of annotations that came with the genome. So this core genome thing is all of the annotation features that came with the genome that we imported. So I can double click on that and see these are all the different tracks that we've got and the number of features that came with them. So there's quite a lot of stuff in here already, and you can import more annotations. So we could have gone into file and import annotations, and we can import from different sources to bring in annotations that didn't come with our original genome. Underneath that are the data sets. So a data set in SeqMonk terminology is a set of mapped reads that came from one imported file. So we've imported two files, 618 and our 619 BAM files, and those then appear under here. The names that the data sets get are just the names of the file that came in, and often the names of files that you bring in are unwieldy and horrible and long. So one of the things that we will probably want to do in here is just to rename these. So if I right mouse click on this and say rename, as it happens, this 618 data set is a H3K4 ChIP-Seq dataset, and the 619 is a K27 dataset, so I'm just going to give them shorter names. But one of the things that we always try and ensure in SeqMonk is that you never lose track of what you've done. So if I double click on either of these, I can see the details behind, and I can see that although it's called K4, it remembers the original name and path of the file that came in, and it records the options that we used for importing it, and then gives me some details about what's in there. So we do track everything as we're going through, and we'll talk about that more later. Data groups and replicate sets, I can't really do anything with at the moment, but those are both ways that you can group data sets together. Data groups are used if you have multiple BAM files that are generated from the same library. So for example, in some cases, you might have done two lanes of sequencing from the same library. You couldn't get enough reads out of doing one sequencing run, so you've done two sequencing runs, but it's on essentially the same library, so there's no distinction between them. And a data group is a way that you could say, well, these three data sets are all the same library, so just merge all of their raw reads together. Replicate sets are used for a different purpose. So replicate sets describe the structure of your experiment. So if you have replicates in your experiment, which we don't in this case, but we will in the other data set I'm going to show you in a minute, then you need to tell the program what samples come from which condition in your experiment. So in a replicate set, you would make a replicate set for each of your conditions, so like a wild type set and a knockout set, something like that, or a male and a female. And then you would add the data sets to the appropriate replicate set so that it would then know how to handle the replicates and what the structure of your analysis was. And we'll see those a little later. Probe lists will start magically doing things when we start quantitating, and probes are the basis for the quantitation methods in SeqMonk, so we'll come back to those in a little bit. Genome view on the top right is pretty boring at the moment. It might do more interesting things later, but for the moment, the only really interesting thing to note in here is that somewhere across all of your chromosomes, up at the top here, is a little red box. And the red box is the part of the genome that you are currently looking at in the chromosome view, which is the bit at the bottom, and the chromosome view is the interesting bit where everything happens. So let's focus on that. So I'm just going to drag this divider up a bit so I can see it a bit more. The chromosome view is a track-based annotation and data browser. So we've got two types of tracks. The ones at the top, these blue ones, are annotation tracks. So you can see by default, we get a gene track, we get an mRNA track, and a CDS track. So we can see the genes, the transcripts, and the coding portions of the transcripts. And underneath that, we have these gray and white tracks, and those are the data tracks, where we can actually see the mapped reads that we imported from our BAM files. Pretty much everything in here is interactive in some way, so as you put your mouse over a gene, you'll see a little label coming up to tell you what it is. If you double click on it, it will open a little dialog to give you a little bit more information about it, whatever it happens to know. Again, all of this is just directly pulled out of Ensembl, so it's whatever Ensembl annotates on it that we can see. Colors in here are important. So the features and indeed the reads all come as either blue or red. Everything that's red is on the original top strand of the genome, so its orientation would be running kind of left to right in this view. Anything that's blue would be on the original bottom strand, so that would be running right to left. Okay, so the colors indicate the directionality, and that's true for both the features, and we'll be able to see in a bit more detail once we zoom in a bit, the reads down here. Let's have a look at some movement controls, because one of the advantages, or the big advantage of having an application like SeqMonk is that you can leap around your genome looking at all the data very easily. So simple things first. If we want to move along the chromosome, there's a little scroll bar at the bottom that we can just click and drag. Okay, so we can go left and right. That can be a little skittish because you're looking at quite a large portion of stuff, so you can use the scroll wheel on your mouse to move a little more easily in smaller chunks. You can zoom in by just dragging a box. So if you just click and drag, if you let go of the mouse, whatever you highlighted in green will then expand to fill the view. So you can zoom in. So I can keep going in until I can see the reads underneath this one little bit of enrichment. And if I want to zoom out, I press the right mouse button, and I can go from looking at a few bases there all the way out to looking at the entire genome. If I want to move to a different chromosome, I obviously can't do that by sort of zooming in and out, but I can either click or drag a box anywhere in the genome view, and that will then move me to a different chromosome so I can see whatever I want from somewhere else. Also, while I'm moving around, I can also navigate in a couple of other ways. So rather than just sort of arbitrarily stepping up and down the genome and having a look at what I've got, I can also navigate either by knowing exactly which position I want to go to. So up here in the toolbar, there is an icon here for go to position. And if I click on that, I can just put in the chromosome in the position that I want to go to, and it will jump me straight there. Or I can put in a location string, so chromosome : start -end, which are the sort of standard strings that you see reported in lots of places, and you can just paste that into the box, and that will also take you to that location. You can also navigate by the annotations on here. So if I click on this icon in the toolbar here, this one that says Find Feature, that will open up a little search box for me. And in here, I can type, well, I can search through lots of things. So the default is that I search through the name of gene features. So if I go and stick a gene name in, so let's stick nanog in and press return, it will then do a search and tell me that there's 3 genes that have nanog in their name. There's actual nanog, and then there's 2 pseudo genes for it. And if I want to go and see that, I can just double click on this line, and it will then show me the nanog gene so I can go have a look at it. If I wanted to do something a bit more broad, then rather than searching through the name of the gene, I could search through all of its information. So on here, if I have a look at the information that's on here, I could search through anything that was in here. So let's say I wanted to search for all homeobox genes. So back in the search, I can say search for homeobox in all of gene features, and that will find me now a whole load of different genes, all of which have homeobox somewhere in the name, and I can just double click on these to go and look at any of these different features. So it's really nice to be able to sort of leap around in these. Other little tricks while I'm in here, I may as well show you. If you have a search set like this, then you can just look at them individually, or you can turn any individual search into an annotation track. So we said that you can add more annotation tracks to the program. This is one way that you can do it. So you can take a set of genes that were defined by a search term and then say, I'm going to save all of these as an annotation track. And I'll call that just homeobox genes. And if I do that, now you'll see a new track has appeared in here, homeobox genes, a new annotation set has appeared up here, and I can see where the homeobox genes came up. Other things that I can change in this track then, because it's the chromosome view is a track-based browser, I can change the tracks that are there. Okay, and the tracks come in two types, the annotation tracks and the data tracks. And those can both be changed with the buttons on the toolbars here. So this one on the left is to change annotation tracks, and the one on the right is to change data tracks. So here, if I click on the annotation tracks, if I decide I don't want the homeobox genes showing anymore, I can just remove them. It doesn't delete them from the project. They're still there and I can add them back later. They're just not showing. But if I wanted to see CPG islands, for example, I can add those in. Maybe I don't want CDS, and maybe I want the CPG islands above the mRNA. You can just pick the tracks you want in the order that you want, and then it will just adjust the track like that. Same thing is true for the data tracks. So if I just click on the next one and say change data tracks, I've got my K4 and my K27, but maybe I want my K27 above my K4. Okay, and I can add those in. Okay, so all of those are just ways that we can configure the view. Actually, let me take CPG islands off just to give me a bit more space. Okay, right. So one of the things I said that was going to be important in here was sort of saying, well, why do you want to load your data into a viewer like this? Why not just go and stick it into a pipeline, go and get it to analyze it? And the main advantage for having a system like this is that you can see what's going on. If there are problems in your data or things aren't doing what they should, in a visual environment such as this, it's really obvious to see what's happened and to be able to spot problems and fix them or account for them. So for example, in here, these are both ChIP-Seq datasets. So the whole point about ChIP-Seq is that you're supposed to be enriching for interesting parts of the genome. So instead of seeing reads just appearing everywhere equally, we should be seeing some places in the genome that have loads of them and some that don't have very many. And just from a static view of part of this chromosome, you can already tell an awful lot about how well these experiments have worked. In that if I look at this K4 data set, I can clearly see lots of places where I have really convincing and obvious enrichment in the data. Okay, so this has worked really nicely. And in the K27 dataset, well, it's not completely flat, but the pattern of enrichment looks nothing like it looks in the K4. So just from taking a couple of minutes of looking through your data, flitting around different parts of the genome, you will very quickly get quite a good impression of how well your experiment has worked, whether samples are doing what they should do, whether they're different to each other, and what the properties of them are. Okay, so that's kind of why we've tended to go down this route, so that here we can see that the pattern of enrichment is super different, but there is enrichment in the K27. It just doesn't look the same as it does in the K4, and there's much less of it. We are looking at raw reads in here. So again, if we zoom right into this, we'll see the individual reads, and we can see that again, they have the blue and the red coloring to indicate the strand that they're on. We've got a few tools that we can use to change the display of the data in these tracks. We'll come back to these controls a little bit later as well when we do some quantitations, but we can look and see what they do with the raw data. So up in the toolbar again, this thing that says data track display options, the one with the little sliders on it. If I open that up, then some things that I might want to do, if I haven't got an awful lot of space to display my reads, so often if I've imported several tracks, I'll see stuff like this where we'll just run out of pixels to put it in. One of the things we can do is to change how big reads are. So here in this raw data display density, we can see we're at low density, which means the reads are all quite big. But by turning that up to medium or even high density, you'll see that the individual reads get smaller, but you can cram more stuff onto the screen. Okay, now for this data set, I don't really need that because we've got plenty. The other thing that can be useful is if you're looking at the directionality of your reads. At the moment, the forward and reverse reads are all just mixed together so that we can get as many reads on the screen as possible. But it can be useful to say, well, instead of putting your reads combined, you can separate the strands so that all the forward reads go on the top part of the display and all the reverse reads go on the bottom part of the display. And that might be a useful thing for assessing directionality. Okay, so we have some choices in here. I say we'll come back to this a little bit later because there are other things that we can do more usefully later on. Now, it's always useful to go and look through your data. You can learn an awful lot just from looking at this. So if I look at this, I can clearly see that I've got nice enrichments in here. I can see that that enrichment comes up on both strands. I can even get an idea that the enrichment is generally sitting at the start of genes. So remember, this is a reverse strand gene. So this is the start. This is a forward strand gene. So this would be the start. Again, here, I'm seeing enrichment around the start of my gene. So without having done any sort of formal analysis, I can already start seeing some things that might be interesting in my project. But a more sort of thorough examination of this is going to require that we actually take a quantitative approach to this. So let's look next at how we can quantitate this project. Quantitation in SeqMonk is done in 2 steps. So the first step is that you decide where in the genome you're going to make measurements. And what you build is what's called a probe set. So down here, you remember in the data view, we had a folder called probe lists, and we said that would come back later. Well, now is the time that we're going to make a set of probes. Probes are simply places in the genome that you make measurements, and the set of probes is all of the places that you're going to make measurements. Probes are defined from the data menu and define probes. And then on the left-hand side, you can see what are called probe generators. So these are just methods of generating probes. So I could do something simple like tiling running probes across the entire genome, which is what I'm going to do in a second. Or I could do something more complex like saying, well, make probes around a particular set of features. Okay, we might do that in a little bit as well. But each of these are different ways of defining a set of probes. I'm going to start off by doing something simple. So I'm just going to put running probes across the whole genome. It gives me a size that defaults to, which is 2 KB. I'm going to make them a bit smaller. I'm just going to put 500 base pair probes over the whole genome, tiled end to end. And if I say create probes, it will then go ahead and make them. It'll ask me what I want to call them. Let's call it 500 base pair tiled probes. Okay, and then in the second step, having decided where we're going to make a measurement, we now get our quantitation options. And in the quantitation options, what the program is going to ask us is, how do you want to use your sequence data to assign a numerical value to every probe in every data set? Okay, and again, there are a lot of different ways to do this. It can be based on the percentage coverage, the number of overlapped reads. It could be to do with quantitating duplication. Lots of different things that you can do. I'm going to do a really simple quantitation. I'm just going to do a read count quantitation. When you select a quantitation method, you get loads of options up. So when I'm quantitating just read counts, do I quantitate all reads or just the forward, just the reverse, those that are on the same strand as the probe, or the reverse strand to the probe? Our probes don't have a strand because they're just tiled probes, but probes from features would have one. So I'm just going to count anything that overlaps with the probe. I'm going to apply some normalization. So I'm going to correct for the total size of my library. So if I have more reads in one of my conditions than the other, I can correct for that. So it's just going to scale up the smaller data sets until they match the counts of the largest data store. By default, it will log transform my counts, but I'm going to turn that off and do linear counts. So I can get a slightly sort of wider dynamic range on this. But again, you can pick whatever options make sense for the type of data that you're going to work with. And I'll press quantitate, and it will now quantitate my data for me. You'll see it goes through. It's quantitating, what's that, 5.5 million reads across 2 data sets, but it does it pretty quickly. So it doesn't take too long to do this. Once that's happened, you'll then see the chromosome view change. All of the data tracks in the chromosome view will split into 2, where the top half will be the raw data that we saw before, and then the bottom half will become something like a bar graph showing the quantitation that you've just done. So in this view now, every probe is kind of a bar in here. So because we made 500 base pair tile probes, all the bars should be the same width and they should cover the entire genome. Okay, some of them you won't be able to see because there's no reads over them, but then the height of the bar represents the quantitation, and the quantitation in this case is how many reads were under there. Now, you can see that the scaling on this isn't great at the moment. The program scales it so that the top of the quantitative scale that you have here is the 95th percentile of the data, but in a ChIP-Seq data, probably fewer than or less than 5% of the genome is enriched, so actually it's not got the scale right. So in the toolbar at the top here, we have a data zoom button, which just lets us alter the scaling on the y-axis in the chromosome view. And here, if we pull this down a bit, you can start to see a better view of the quantitations that we have in here. So now you can see the quantitative view of what we were looking at just sort of qualitatively before. And we can see nice sharp peaks in the K4. In fact, if we take it so those are all in there, we can see that the enrichment is much higher in K4 than it is in K27. These go down, but if I sort of adjust it a bit so they're both in there, some of these are saturating, but we can see the enrichment in there. So now we have a nice running quantitation of what we've got in here. A few more options then. So now that we're displaying the data in our chromosome view with both the raw data and the quantitation, firstly, we should check that the two match. So we expect that we get high values where we get lots of reads, and we can clearly see that that's worked in this case. But if we'd messed anything up, it's really easy to spot it because you'll immediately see that the quantitation doesn't appear in the right places or doesn't match with the data in the way that it should. So again, it's really easy to spot if something has gone wrong. In our display preferences, we've got a few other things that we can change. So here by default, we are showing both the reads and the probes. That's fine because we've only got 2 tracks on here. If we had a lot, then maybe we haven't got enough space to put all of them in. So we can choose to go back to just looking at the raw data or just looking at the quantitation, or we can put both in. We can change how the quantitation is shown. So the default is that it's this kind of bar plot. We could do it as a line, so we can have it on there. We can even just do it as points, so we can scatter those across, and blocks, but blocks are, yeah, create like a heat map underneath. So again, you can pick the representation you want. You can choose the colors. So your choices are that you either have a color scheme which reflects the quantitation. So when you do the data zoom, you can see the color scale here, and the bars are colored based on that. But you can just turn them into fixed colors. So you can say, well, make all of the first ones blue and all the bottom ones red. Okay, and it will just put them in a solid color, which can be a bit nicer if you're trying to make figures out of this. And if you are going to do it as a gradient, you can pick different types of gradient colors to suit your mood. Okay, but those are the choices that you get. Now, we can see the quantitations in here. If we select a data set, we can also see the quantitations now appearing in the genome view. So if we wanted to see what that looked like on a wider scale, we could do this so we could see where the most enriched portions were across the entire genome. So that will also be useful, so we can project stuff on a whole genome level. But probably more usefully, once we have quantitations, we can then go into the plots menu here, and there are tons of different plots that we can use to explore the quantitations that we've got. So let's have a look at a few plots that allow us to see interesting things in our data. So in the plots menu, I'm going to try a few different things. If I start with this K4 plot, and I can select a histogram of the values that we currently have for the quantitation. Okay, so this is a probe value histogram, and it looks a bit weird. And it looks a bit weird because we have virtually all of our data crammed in this little thing next to 0, but somewhere up here there must be at least one probe that's got values of sort of over 20,000 in here. We can zoom in on the bottom end of this plot to look at it in a bit more detail, and we can put it on a log scale to try and look at the distribution a bit better. And what we can see is that nearly all of our probes have values that fall kind of under 500, but then there are a few weird outliers that are super huge and will end up sort of skewing the distribution. If I look at this in a different way and I do a scatter plot, I can do a scatter plot between my two conditions, and that also is going to look weird because of these sort of stupid outliers on here. So most of the interesting data is crammed down the corner here, but these outliers are messing things up for us. So looking at using the visualization will help us to figure out that strange things are going on, but now we can also go ahead and fix it. One of the other sort of fundamental concepts in SeqMonk then is the idea of filtering probe sets. So if I look back in my data view, I've got my 500 base pair tile probes here, of which there are like 5.5 million. But I can reduce those. I can get rid of ones that I think are uninteresting for whatever reason. So in this case, what I'm going to do is to get rid of all of those super highly quantitated probes, which are probably all artifacts, so that I can look at the rest of the data in much more detail. You reduce your sets of probes by applying filters. So there is a filtering menu, and the filtering menu has lots of different methods by which you can choose to select some probes and reject others. So again, I'm not going to go through all of these, but let's look at a simple one, which is to filter on the current quantitated value. So if I say filter on value and individual probes, I can say, right, when I looked at my histogram, I saw that all of the data was basically under 500. So I'm going to say the value must be between, well, anything and 500, and it's got to be in that for exactly 2 of my 2 selected data stores. So both of them must be under 500. I run the filter. It gives me a slightly reduced, so I've still got nearly 5.5 million. There's not many of these. So I will say, I will call this like sensible coverage or something. And now I will see in my probe lists that I've got a little plus next to this, which I can expand and see that I have a child list now, which is a reduced subset of that original list. And I can apply filters sort of as many times as I like. So for example, if I wanted to take this and then say, well, I only want to look at the probes that are on chromosome 1, I could go back to filtering and say filter by position, and then say, well, I only want things that are on chromosome 1 and at any position, I don't care. Now I get a much smaller list of probes and another level of filtering where I can see that. When you select a probe list in here, only the probes in that list will be shown in the chromosome view and the genome view. So if I click on the chromosome 1, you'll see all the probes on the other chromosomes all disappear. So we can just see chromosome 1. And here I'm only looking at the ones with sensible coverage. Also, when I do my plots will only be drawn off the list that I have selected. So if I go back and do my scatter plot again now, I can see, oh, let's do that the other way around, K4 against K27. I can see a much more sensible view now because I've limited myself to values of less than 500. So now I can see my data and I can see the patterns that are going across in this. So if I look at this plot, what I can see quite nicely is that I have a bunch of probes down here that basically have no signal in either. Okay, that makes sense. I've got a set of things which are very high in K4 and very low in K27, and not very much that's high in K27 and low in K4. And then some things that just have unusually high coverage in both. One of the nice things about this is these plots, again, are interactive. So if I can see stuff like this, where I can see these highly different probes, I can just put my mouse over them and double click, and it will take me to that point in the genome. So I can say, well, where I get something which is a very K4 specific peak, does it look like a real piece of data? And yeah, that looks fine. Look, I've got lots of stuff all appearing in one place. I really don't see anything in the K27, and that all looks good. And it's really easy to just keep picking things out of here, checking, seeing if you believe it, or if you see something weird that might be a problem with your analysis that you would want to fix. We can also try the same thing the other way around and go and pick some things that are high in K27. So if I do that, here's an example of something which is high in K27 and low in K4. That one doesn't look too bad, except that in this case, I can see this is over a gene called SFI1. People who've worked in bioinformatics for a while, you kind of learn certain genes that smell bad, and SFI1 smells bad. It's a gene that's on the end of the chromosome, and it's one that you get variable duplication of the end of this chromosome. So actually the copy number of this varies hugely. So actually, although there is a difference here, it's probably because the genomic copy number is different, and you can see that it's on the end of the chromosome because there's the end over there. Let's have a look for anything else that's in there. Oh, that's another SFI one. What's this on? It's on chromosome 11. So let's see if we can find something that's not on chromosome 11. There you go. That's on chromosome 4. Okay, so this is one where we get a higher signal in the K27 than the K4. But when we look at the data underneath, it doesn't look right. So instead of being a nice broad set of enrichment, it's kind of this weirdly discontinuous set of individual positions where there's way more reads in here than there are in here. But it doesn't look right. So again, this will be really hard to identify if you were doing this in a sort of pipeline methodology, but in a graphical environment, it's really easy to see that although the numbers say that this is different, something can't be right here. Okay, so those are the sorts of things that we can look at in here. Let me show one other thing. So the plots that we've looked at so far in here have directly plotted the quantitation that we got. So we looked at the histogram of the quantitative values in a scatter plot comparing two values. One other type of plot that you can do is a summarization plot. So if we look more generally in this data, let's just pick a chromosome 1 to go and have a look at. We can see that if we look where the peaks are in here, that there's a suggestion that those peaks all seem to turn up around promoters. They're all at the starts of genes. So whenever we go in here, look, this is at the start of this gene. This one is at the start of that gene. So it's nice to be able to sort of get some sort of summarization to be able to see if that's true generally. We've got probes tiled across the whole genome. So one of the other plots that we have in here is what's called a quantitation trend plot. And if I do that with the current probe list, what I can do then is to say, well, build me a summary of the average signal around, in this case, an average gene. Okay, so I can say, go and design this plot around genes and do the gene from sort of 10 kilobases upstream of the start of the gene through to 10 kilobases downstream of the end of the gene. And if I do that, it will build me one of these little metagene plots here, where I can clearly see that in my K4, I see huge enrichment at the start of the gene that falls through the body. And in K27, I see a little bit of enrichment. It does still follow the same track, but obviously to a much lesser degree. I can also see that inside the gene body, the K27 is sitting a bit higher than it is outside, whereas the K4 is much less clear that there's a distinction outside. So maybe there's some suggestion that you have enrichment through the genes in K27, whereas they're very specific to the promoter in K4. Okay, so all of these kinds of plots help you understand and explore your data before you get into any sort of specific kind of analysis or selection of points. One last thing I'm going to show you in here, and then I'm going to switch to a different data set for the last few minutes to illustrate some other things. In a graphical environment such as SeqMonk, one of the problems is keeping track of what you've done. We obviously recognize that this is a problem. We want to do reproducible research. We want to be able to track what we've done and be able to report exactly so that we can do it again. And SeqMonk does this. Up on the toolbar at the top, there is a thing that launches what's called a history. Now, I think this might be one of these windows that disappears. So I'm going to quickly switch across and show you the history window that opens up. A history is a way to record. Do it. Let's try that again. There you go. Record what it is that you've done in the program. Now, what you can see here is the barest form of this, where every operation where you've added data sets, you've renamed some stuff, you've quantitated, you've done some filters, each of those is automatically recorded with all the details of what you've done in here. So here is a list of the operations that you've got. You can then annotate this, so you can put sort of titles and subtitles and bits of text and embed other things into here so that you can create nicely formatted reports. If you're interested in this, one of the earlier Bitesize Bioinformatics sessions last week was on the use of history. So there's a whole talk you can go and look at on our YouTube channel, which goes through all of the stuff that you can do in here. But these are basically the way that you can record what it was that you did. Right, for the last little bit that we're going to do, I'm just going to switch back again to SeqMonk. And I'm going to switch to a different data set. So in here, once you've finished with what you've done, you can save your project. So if I say save project, and I will go into here. SeqMonk projects save to a single file, and that file has all of the data, the quantitations, the probe sets, the filtered probe sets, everything is recorded in there in a single file, so you can give that to someone else or you can put it onto a different machine and everything moves with it. You don't need to track anything else with it. I am going to open up a different data set to be able to illustrate a couple of final features, and then we will be done for this. So I'm going to open a project, and the project that I'm going to open is in my example data and is going to be this RNA-Seq example. So the reason I'm opening this is because this is a project that has more data in it. So this has 3 different conditions and 3 replicates for each condition. So this is going to allow us to firstly look at how to handle the replicate sets that we didn't show you before because we didn't have replicate information. And we're also going to get a chance to actually show you how to run a statistical analysis, a very trivial statistical analysis in this case, but to show the principle for how that works, and to show a couple of other little features in here. Okay, so here you can see a larger data set loaded in. we have 9 samples in here. These are RNA-Seq, so they're measuring transcription. So again, just from a visual point of view, if I zoom in on here, I can immediately see that the reads in here are sitting over the exons of transcripts. So if I wanted to be sure that my data was what it was supposed to be and was behaving nicely, you can spot problems really quickly by looking at this. We also provide some mechanisms to detect problems more automatically. So down here, for example, we have a specific plot for looking at particular QC metrics within RNA-Seq, which again, I'm not going to go into for today, but just to illustrate that things like this are built into the program. So if there are sensible things that you can summarize for a particular data type, we've tried to add those in. So for example, here is a QC report for our RNA-Seq, so we could spot if we had samples that were doing weird things. Okay. Now, the thing that I need to do in this data set is I need to describe the structure of the data set. So I have 3 ISH samples, 3 MCF7 samples, and 3 T47D samples. But at the moment, I haven't got any replicate sets. So I'm going to build these. So under data, I can say edit replicate sets. And to do this and to build the structure of my experiment, I simply make new replicate sets. I'll make an ISH set, select it, and then add my 3 ISH samples to it. Same for the MCF7. Oops. And finally, the same for the T47Ds. Okay, so now I have 3 replicate sets for my 3 conditions. And when I go to the track-based view, I can remove all the individual samples, and now I can add in the replicate sets. When you put a replicate set in, you can do one of 2 things. So the default view is that you look at the combined reads from all of the data sets in the replicate set. So I can just see 3 tracks, 1 for each of the 3. And if I had quantitations, I would be looking at the mean quantitation across all of the replicates. I can, in the data track display, one of the options there is that I can choose to have my replicate sets either compressed or expanded. And if I expand it, then I can see all of the individual tracks, but they're always sort of firmly grouped together so that the replicates for one condition will be placed together. I'm going to do a quantitation with this. For certain data types, there are sort of built-in quantitation methods for them. So what we showed you before with defining probes and then quantitating the probes is the most sort of general way of quantitating. We have some shortcuts which are called quantitation pipelines. So that, for example, for RNA-Seq, we have a pipeline which is designed to quantitate RNA-Seq data explicitly, and it's a read count quantitation, but with certain normalizations and corrections built into it, which again, I'm not going to go into today. We'll do a session on RNA-Seq later on. But it means that in a single step, I can define all of the probes and quantitate them and get everything that I need out of this. It'll take a little time because this is going to be 9 samples that it's quantitating, but again, it's pretty quick. And then we see the same sort of view as we saw before, where we have a bar now over every gene, so we're quantitating at the gene level, and the height of the bar represents the expression level of that gene. I'm going to show a couple of other quick analyses in here just to illustrate some different points out of this. So we've got a bunch of quantitations in here. One of the plots we can do on here is to check the normalization. So this cumulative distribution plot is a way of plotting multiple distributions together. So here we can see how well matched all of our 9 samples are. They're reasonably well matched. We can also cluster our samples. So we should have 3 groups in here, ISH, MCF7, and T47D, but it's really nice to be able to use the quantitations of your data to show that the samples that look most similar to each other are the ones from the same condition. And we have a number of ways that we can do that under this data store similarity menu. I'm going to do a PCA on this, and this will illustrate another aspect of SeqMonk, which was that at the start we set up a link to R, And at this point, this is one of the functionalities in SeqMonk that goes out to R. So when it does that, you'll see a little R session open up. It runs a bunch of R code, pulls it back into the program, and now I can see a PCA plot that was calculated in R, but off the data that was in SeqMonk, and then visualized within here. I can see my samples in here, so I can see that actually I've got 3 nice groups of samples. I can highlight the replicate sets that I had before, so that I can see that all of the ISHs are these blue ones, all of the red ones, the MCF7s, and all the green ones are the T47Ds. Okay, so they all look really different to each other, and again, it's a nice reassuring thing to be able to see that my data is behaving the way that I would like to make it behave. I'm going to finish up by doing some filtering then. So I'm going to run a statistical filter, and I'm going to run one that will allow me to find things that are different between any of these replicate sets. Statistical filters are just part of the filtering menu. We have lots of different statistics under here which are appropriate for different types of data and different types of quantitation. For RNA-Seq, we would generally start with proper count-based quantitation. So we would use things like DE-Seq or EdgeR. I'm not going to do that now just because it takes a little bit longer than I want to spend on it. So I'm going to do a more simple test, which will give us some interesting results on here. So I'm going to do what's called an intensity difference test. And this is just going to find the most changing genes between those different conditions, but it's more the principle here than the test itself that I care about. I'm going to compare all of my replicate sets to all of the other ones, so anything that's different anywhere will come out of this list. And if I run that from my 31,000 genes that I started with, I get a few hundred, which are the most changing in here. And if I now open up a scatter plot, so we saw scatter plots earlier, I'm going to pick a pair of samples on here. You can again see how different these things are. You can see which genes are which, and if you double click on them, you can go and see how they behave in the data. So all the same sort of stuff that we saw before. There you go. There's something that really looks different. And now we can highlight on the results of our filter. So we can see the things that are changing. Not all of them were changing in this comparison because they could have changed they could have changed in the comparison to the MCF7, but we can see that all of the things that fall a long way from the central point are represented. So most of these plots have ways to highlight sublists and show interesting things from there. Okay, one last thing then to show you for the analysis. Just now we have a slightly more complex data set. From this set of interesting things that could be interesting in any of our 3 samples, I'm going to do a different plot. This is a heat map plot. So this is then going to cluster my samples. I'll just change the colors a bit and change the scaling a bit. But now it means that I can see my 3 different groups, and I can see the expression profile across them. If I just highlight the lists, they're pretty obvious already, but let's just make them a bit more explicit. There are my 3 groups, and I can see that some genes here are, so anything that's green is highly expressed, anything that's purple is lowly expressed. Some genes are expressed specifically in ISH, some are specifically in T47D, some are specifically in MCF7, and various patterns come out of this. This again is interactive, so this interactivity is a key sort of thing within SeqMonk. So I can double click on any of these to go and see what that gene is doing. So there you go, there's an ISH specific gene, disappears in the others. Let's look for a T47D specific gene. Oops, lost it. There you go, so there's a T47D specific gene. And finally, let's try and find an MCF7 specific gene. Okay, so you can see all of these different patterns coming out. So various ways in which we can illustrate this. The last thing I'm going to do from this is that we can use this plot to break apart all of the different clusters that we've got. So if you look on the left-hand side, you can just about see these little black lines poking out from here. These delimit the parts that SeqMonk is deciding behave similarly. So this is one cluster up here, then there's another little cluster there, a bigger one here, a bigger one there, and a bigger one there. So if I save those clusters, what I'll now get is a set of genes which should all now behave the same across my different samples. So if I go back and do my hierarchical clusters on one of these again, you should see that as before we saw variable patterns, but now we see a nice consistent pattern across the whole set. And if we want to have one last look, if we do a line graph for all of, oops, sorry, didn't mean that. If I do a line graph for all of these lists, I can select all of them and plot them all at the same time. And if I highlight the groups on, it means that I can now extract out all of these different patterns with all of these different sets of genes, and those I could then feed forward into sort of gene set enrichment or whatever downstream analysis it was that I wanted to do with this. Okay, that's all really I want to show you. I mean, it's the idea with today is to give you a feel for the sort of way that the program works and the kind of things that you can do. Obviously, we can explore this in more detail. The last little thing I'm just going to show you is how to export things out of the program. So I'm going to export two things. I'm going to export a report. So if I had my interesting cluster of genes here and I wanted to take that out and put it into a paper or to do more work on it outside of SeqMonk, then I can do a report from that. So under the reports menu, I can do an annotated probe report, which means I'm going to get a report on every probe in this list, so 77. And I'm going to annotate these. The probes are all named after the gene that they're measuring, so I'm going to put more details about the gene based on a name match to that gene. Loads of options that I'm not going to go into now, but the principle is that you can see that you get a nice tabulated report which has the probes, where you measured it, the corresponding gene, and loads of details and descriptions around that. All of the quantitations that you've got, and you can add in things like the p-values from statistical tests and anything else that you want to annotate on here. And these again are interactive. You can sort on any of the columns here, and you can double click on any column in here to go to the corresponding data in the main view. The last thing I want to show then is extracting images from here. So if I do another plot, let's do a beam plot from here. Okay, so this is for my cluster. This was a cluster that presumably was low in ISH and high in MCF7 and T47D. Any plot that I have up here, I can save. So saving a plot, I can save it to a number of places. I can just copy it to the clipboard. I can send it to the Vistory that I briefly showed you before. I'm going to send it off to a file. And when saving to a file, you can either save it as a PNG, which is effectively a screenshot, or you can save it in an editable format. So here I've got an SVG file, which is a vector graphics file, which will mean that I can have a file exported out of here, which I can then load into another program. So here I'm just going to quickly switch across to a local copy I've got of Inkscape. Inkscape is a free vector editing program. And the file that I just saved, I'm just going to open up. If I can find it, example, there's my beam plot. And if I open that, oh, Inkscape changes size on me. Don't do that. There we go. Inkscape then will be able to play around with this data. It's all in an editable form so that you can go around, you can start changing colors, you can zoom in, play around with sizes and fonts and do whatever it is you want to do. Everything in here is completely flexibly editable, so you can use the program for generating things which will form the basis of publication quality graphics that you can run in there. Okay, we've gone a little bit over the hour, so I'm going to stop there. Let me turn off the sharing. Obviously, that's a lot of stuff to go through. On our Babraham Bioinformatics web pages, there is a lot of documentation for how to use SeqMonk. We have courses that run on particular data types that will go into more detail from that, and there'll be more of these events that will go into more detail for than we've gone into today. But hopefully this has given you a feel for the sort of things that you can do and the general way that the program works. I'm going to stop the session now. If anyone's got any questions, I'm quite happy to take stuff, so I'll just open this up now. If you want to ask a question, just unmute yourself and ask. So yeah, we'll take anything if anyone's got questions. Veronica.
Hi, thank you. I'd like to just go back quickly to the data groups and replicate sets, if that's okay. So there are a few tools that allow us to merge BAM files from things that were sequenced on different lanes. Is there a particular advantage to doing or not doing that and then putting things in data groups?
It's more efficient if you can merge them in the BAM files before you bring them to SeqMonk. So if you have the option of doing that and you've got the ability to do that, it'll be slightly quicker if you pre-merge them and then import them. But obviously, if you've been given the BAM files and it's all done and you don't have that ability yourself, then you can use data groups to put them together in SeqMonk. But that will be slightly slower than dealing with a data set that's been merged before it came in.
Right, thank you.
Okay. Okay, if there are no other questions, then I will finish the session now. This video will go up on our Bitesize Bioinformatics playlist on the Babraham Bioinformatics YouTube channel sort of later this afternoon. So if you've got any other questions, you can add them onto there. You can, yeah, if there's anything else, you can also go to the Babraham Bioinformatics website, and if you look on there, the contact details for all of us are on there. I hope you found that useful. There's another Bitesize Bioinformatics coming at 3 o'clock this afternoon, where Felix is going to be talking about Nextflow pipelines, and you can look on the Bitesize Bioinformatics web pages to see the details of all the future talks that we've got. So hopefully we'll see some of you at those as well. Okay, thanks for coming along today.